Dr. Wild. He is heading the molecular laboratory in pathology. And his talk will be next-generation and liquid biopsies.
So thank you very much for the invitation. I would like to give you more the view of the daily user of what we have heard in the first talk of this session. And I will give my talk together with Alessandra Curioni. Now, these are my disclosures. What's the big goal? The big goal is precision medicine, and I will not talk about all this today. Today, I will talk about the molecular barcoding. However, the molecular features of a tumor, this is just one part of the patient. We all know that. There is, of course, the pathology images. There is the health records, the drug history, the lifestyle, and so on. This is a big project that we are now developing together with people from ETH and the University of Zürich. And again, today I only focus on the molecular barcoding. So now we have heard that the Cancer Genome Atlas has done a lot of barcoding already. And the question is, can we use this barcoding from the genome as if it was a bottle of water? Like if you go to the supermarket, you take a scanner, boom, you know what it is, without even looking at it. And I think the answer is almost yes. So this is the barcode again that we have seen now from the former speaker. And I would like to just focus on one gene now, KRAS. Now, as you can see, KRAS is mutated quite often in lung adenocarcinoma. 30% of the pie chart that you have shown is KRAS. It's mutually exclusive with most other mutations. So you're either on the KRAS or not KRAS. Now, can we use and analyze all this from liquids? Yes. The answer is yes. So in the future, I think we will just take blood from patients and generate the same kind of pie chart and use this even to follow the patients over time and not just have a molecular barcode from the diagnostic, from the initial diagnosis, but also during treatment. And then also identify resistance mechanisms. But coming back to KRAS. Now, it is very interesting that from the whole genome, and you are doing whole genome sequencing for patients, that is really very, very interesting. But actually, it only takes one single nucleotide to be changed to really activate this protein. And that's what we have to look for. So it's only one little nucleotide that then leads to a mutated protein, and that is powerful enough at the end to kill the patient in a simplistic way. Now, I will not go into detail about next-generation sequencing, but I would like to show you the problem that we are facing. What we get from these kind of sequencers at the end is a lot of reads, as we call it, a lot of nucleotides and information. And these reads and the currency that is used for such a sequencer is millions of reads. So let's say such an ion torrent machine would come up with 5 million reads in a good run. The very new sequencers that have been now on the market, or that are on the market since 2 days, they have 10,000 million reads, the NovaSeq. Yeah? And all this is done on these chips. I even brought some chips for you. You don't have to look at this, please. And one such chip will produce 5 million reads. We will then compare the reads with a reference genome, and at the end what we get is the percentage of mutations for each target region, right? So 50% of mutated reads for KRAS, for example, exon 2, or 25% EGFR. Now, this was the very first report that I ever signed out doing next-generation sequencing. Please don't laugh, it's only a few exons. But it's ages ago. It was 2013, and it was paid by health insurance. So this is also quite unique to Switzerland, that the health insurance paid for this analysis. And you see that we found this KRAS mutation in 40% of the reads. We had a sequencing depth, a coverage of 3,000. Now, doing diagnostics requires completely different measures than research. I would, in a research setting, a coverage of 150 for an exome is good, but for diagnostics, it's never enough. I would like to have 500 or even higher coverage, but then it gets way too expensive. All this costs us less than $1,000. So it is affordable, and the question is it good enough? And for this, we started a Swiss-wide trial. At the end, it was a European-wide trial, but you can see the different institutions within Switzerland and also Germany, France, Austria that participated. And what we did, we sent around 16 different tumor samples, so the isolated DNA from lung and colon cancer. And what you see is at the end that the sensitivity of what we did all over Europe is 99%. So that was a very, very good result. And that has helped us to get an accreditation for the lab and to convince the health insurance to reimburse it. Now, I would like to talk a little bit about an assay that we are now using. This is the team that does all the diagnostics in the lab. The biggest challenge that we are facing is when we go from single genes to analyzing a lot of genes, that is almost impossible to follow the literature. That you need a database that helps you to, at the end, sign out the case. There are a lot of different initiatives, the CIVIC, OncoGB, the Global Alliance for Genomics and Health. There are also some commercial ones, the OncoPortal, OncoMine Knowledge Base, and this is what I would like to talk about now. Now, Scott Tomlins and his group, what they have done, they have collected a lot of data from exomes, from copy number arrays, from RNA sequencing initiatives, and they have put all this into a knowledge base, into a database. And this database is available for us, and at the end, this is the smallest common denominator for the solid tumors. So what you see here is the 1% variant map. Whenever there is a little dot, then in this little gene or in this site on the genome, there is a mutation, a driver mutation in at least 1%. So the question is there an assay that covers all these amplifications, deletions, fusions, and mutations in these tumors? The answer is yes. It's the OncoMine assay. And there is a small and a larger part, and the small assay, that's the one that we use, is a DNA-RNA combined assay that covers the hotspot of these genes and copy numbers, so amplifications and gene fusions. At the end, you can do it all in one. Yeah. And in total, it's 52 solid tumor genes. Now, this is the order sheet, and that's how you can order it from us. Yeah? And the question is, does it also work with cytology specimens? Yes, it does. We did a comparison from histology versus the cytoblocks that Alex Soltermann spoke about, and we even detected new, also druggable mutations and fusions by within this comparison that had an influence on the treatment of the patients. So it is very, very good, even with very low tumor percentages in these effusions. Now, to give you an outlook on liquid biopsies, there are new assays now available, and they will be available in our lab in February this year, so quite soon, that have not a detection limit of 5% or 4%, but 0.1% from plasma. So we can look for T790M by using NGS if we are able to have 20 to 30 nanograms of DNA from these standard plasma sample, the K2 EDTA plasma sample that you all use in the clinic. And we can look in this isolated DNA for around 150 hotspot alterations. There is another outlook. This is now also up and running. It will be available quite soon, in a couple of weeks. We will offer a fast track, so you will get, for single genes, a result within 2 hours. So it will be a same-day sequencing service. You send in your blood sample, and the same day I will call you back and give you the result. This only works for a few genes, but I think this will become better and broader in the future. Now, we have to select the best assay and platform, of course. This is an ever-growing and developing field. We will have to do a lot of proficiency tests to really control what we are doing. And we have to define standards for reporting. There is a weekly and 4-weekly event, the Cancer Center Zürich, or organized by the Cancer Center Zürich, the Molecular Tumor Board, and I would all like to invite you to join. Now, we are decided to present 2 cases. I will present a very short first case, and then Alessandra Curioni will present the second case. The first case is a 53-year-old male patient with lung adenocarcinoma. He had this typical exon 19 deletion. You see here, this is a small biopsy from his pleura, and they performed Sanger sequencing and found this mutation. What happened? The initial diagnosis was in 2015. The patient was put on afatinib, and what you can see is that he responded quite well. But unfortunately, in 2016, so last year, he had or developed an oligoprogressive disease, and then I got a liquid biopsy. So that was the first time that I got to know this patient, or at least parts of him. So I received a plasma K2 EDTA vial, 10 milliliter. And the question is why? So the question on the order sheet was T790M. You all know that. There are different resistance mechanisms for patients that are treated with EGFR tyrosine kinase inhibitors, and it's mostly T790M, but there is also MET amplification, HER2 amplification, EMT, small cell cancer, and a little bit that is unknown. But all this we can detect with next-generation sequencing, with this Oncomine focus assay. Now, this is what the report looked like. Again, you see it was a liquid biopsy. This was the initial diagnosis, and this is what was known from the Sanger sequencing, and this is what I found. So I found the mutation, the deletion, but I also found the T790M, and the most important for you is this here. You can see that I had a mutation ratio of 0.2, so 20% of the reads were mutated. Yeah? We had a coverage of almost 4,000 here, and 11% of these 4,000 reads also show the T790M. So there must have been a lot of tumor DNA in this blood sample. And then what happened? At the end, the patient is not yet put on osimertinib because he's still responding, but this is then the next step for him, or the next opportunity. And you all know this publication because it's known that it will be very effective, at least in part of the tumor. Now, this is what the Oncomine knowledge base report looks like. So we upload the VCF file into this database that I told you before, and this is then what you get. And I will not show you the full report, but just highlight you that you will automatically get information on all of these trials, on all the labels and FDA-approved drugs, and including osimertinib. Yeah? It will be the first hit. So now the second case. Alessandra.
Thank you very much. Ladies and gentlemen, dear colleagues, it is my pleasure to share with you this case. I would like to say that we decided, with Professor Wild asked me to present this case because it's true that the oncologist is there to take the decision, a tumor board, but we take also all the responsibilities of the treatment we start with these patients. And this is a case of a patient coming from Italy who wanted to have a second opinion, wanted to start soon with treatment by an adenocarcinoma of the lung, EGFR mutated in stage 4. You can see here an example of one of the bone metastases. So we started a treatment with erlotinib, and this was July 2014. The patient had then responded to treatment for about a year, and then in August 2015, he had a progression of disease, as you can see here, as well as in the bone. Well, what we did at that time, we contacted the colleagues of pneumology, Dr. Franzen, who did a bronchoscopy and biopsy, and we could detect the EGFR mutation as well as the MET amplification. Based on the literature, we started to treat the patient on top to erlotinib with crizotinib due to the MET amplification. Please keep in mind that all the diagnostic procedure made by this patient, as well as all the treatment, were paid by the patient himself, because in Italy, all this treatment would have not been covered by the insurance. So what happened is that the patient had a progression, and this was summer last year. At this time point, we didn't know if a lung biopsy would have been enough. So what I decided to do was to do at the same time liquid biopsy and lung biopsy in order also to understand and learn more about this case and this tumor. So in the liquid biopsy, what you can see here, there was the previous EGFR mutation, the T790M mutation, the MET amplification, and an EGFR amplification. At the same time, the lung biopsy showed the EGFR mutation already known, the EGFR amplification as in the liquid biopsy, and ALK translocation, and the MET amplification. So we had long discussions, I have to say, at tumor board, and I remember I painted all the walls of the pathology department to try to understand and figure out what's going on with this case. So at the end, the final treatment decision was based on the fact that the patient already had erlotinib and crizotinib, and by these EGFR, MET, and ALK would have been covered. The only new mutation was T790M, and therefore we gave to the patient osimertinib. This was started probably also with some delay, and probably also due to the disease progression, which was very fast. The patient unfortunately died. So we are not able to say if the treatment would have worked, as it has been given just for about a couple of weeks. I think that this is the clear case where we can see that we can discuss for a long time at tumor boards. We have to face, however, the limits that we still have about treatment, but I think we still need to keep with the same time liquid biopsy and tumor biopsy in order to understand more and maybe give some more chances to our patients. Thank you very much.